Hi, I'm Mr. Williamson, and this is the second video in 5 about DNA and protein synthesis. In this video, we're going to talk about the structure of DNA. Make sure you're taking good notes. Again, Cornell style works for me, but whatever gets your plane up in the air, you can use it. Just make sure you're taking good notes. If you have any questions, please make sure you ask about them in the Edpuzzle questions or in class. You can pause this, and you probably should along the way a couple places. Make sure you pause it. You make sure you get the information down. Here we go. To start with, before we even get started, let's review what we talked about the first video. Watson and Crick, the guys who discovered the structure of DNA, they knew quite a bunch of information about DNA. They knew it was control the cell somehow, but they thought it was a protein. They fixed that. They knew that it was a large, long, thin molecule. They knew that it had to do 4 things. Had to be able to store information, you had to be able to read it, copy information, copy itself exactly, and be able to make mistakes. They knew that it had a sugar, a phosphate, and there were 4 nitrogenous bases. They knew all that. What they came up with in their model, and they knew the size of those things, what they came up with their model was the double helix. All right? The double helix consists of a double strand of alternating phosphate and ribose sugar. Just to say you're up the outside of the DNA, I'm going to show you some diagrams. Outside of the DNA is alternating phosphate, sugar, phosphate, sugar, phosphate, sugar. They make up the backbone of the ladder. So you have the phosphates in there. Here's a phosphate, sugar, phosphate, sugar, phosphate, sugar. So it's alternating phosphate sugars all the way down the backbones of the DNA. All right? The strands on one side of the DNA run opposite directions. It's called antiparallel and complementary. This is a little bit above what I'm going to expect, but I'm just explaining it to you. All right? You'll notice that in this, on the right side, the pentagons point down. On the left side, they point up. That's antiparallel. All right? The structure is the same, but they run different directions. Complementary means that's what's on one side over here complements what's on the other side over here. What's on one side here complements what's on one side there. There's a very specific order. What's on one side over on this side is determined by what's on that side, or vice versa. It could be what's on this side over here is determined by what's on that side over there. But they complement each other. Antiparallel and complementary. Between the strands are the 4 nitrogenous bases. You're going to need to know these, all right? So we have adenine, we have thymine, we have guanine, and we have cytosine. You're going to need to know the structure of these, and I've got a way to help you do that. Adenine is a purine. It's a classification of type of chemical. And adenine is a purine. It has 2 rings, and again, like all the rest of the videos, you do not need to know the structure like all the carbons and nitrogen things. All right? You need to know that it has 2 rings, and it forms 2 hydrogen bonds. So it has 2 rings. This is one ring here. Here's a second ring over here, and it forms 2 hydrogen bonds. So it's double ring, double bonds, and the category is a purine. Adenine. It's a purine with 2 rings, 2 bonds. Guanine is also a purine. It's also double bond, but it forms 3 rings. So we have, again, the first ring, we have the second ring, and we have 3 bonds. Guanine is a purine, just like adenine. It's 2 rings, but 3 bonds. Cytosine is not a purine, it's a pyrimidine. It's a single ring, forms 3 bonds. Cytosine, a pyrimidine, 1 ring, forms 3 bonds. Thymine is also a pyrimidine, has a single ring, forms 2 bonds. And this is a little addition to this one. We don't find it in mRNA. And you don't know what mRNA is right now, but we're going to talk about that later. You don't find it in mRNA. It's replaced by a nitrogenous base called uracil. Which brings us to our last one, also a pyrimidine. It's also single-ringed. And it also forms 2 bonds. I assume it's off those. Okay? You don't find it in DNA. There is no uracil in DNA. You only find thymine in DNA. You find uracil in messenger RNA. You do not find thymine. You do not find thymine in messenger RNA. You do not find uracil in DNA. Okay. Here's the way they pair up. Adenine always pairs with thymine. Cytosine always pairs with guanine. So if you have adenine over here, you're going to have thymine over there. If you have thymine over there, you're going to have adenine over there. If you have guanine here, you're going to have cytosine there. And if you have cytosine there, you're going to have guanine over there. They always pair up. Adenine with thymine, guanine with cytosine. Make it easier. A's with T's, G's with C's. And I'm going to make it even easier. If you don't even need to remember the letters, I mean, at some point, there's going to be the word in front of you. Think about it. I know you're thinking of the test. How am I going to remember all this? Someplace on the test, there's going to be an adenine, a thymine, a cytosine, and guanine. That word's got to be there somewhere. Okay? So how do you remember what pairs up with each other? Straight lines, crooked lines with crooked lines. And you're like, Williamson, what are you talking about? Straight lines with straight lines, crooked lines with crooked lines. Straight line, crooked line. A's with T's, G's with C's. Always. So the whole molecule is a phosphate, a sugar, and one of the nitrogenous bases. That is the monomer of a nucleic acid, is a nucleotide. You remember monomers. We go back to the second unit. Remember the monomer of a carbohydrate as a saccharide, the monomer of a lipid is a fatty acid, the monomer of a protein is an amino acid. Well, the monomer of a nucleic acid, DNA or RNA, is called a nucleotide. And a nucleotide consists of a phosphate, a sugar, and a nitrogenous base. This down here is a nucleotide. It has a phosphate, a sugar, and a nitrogenous base. And in this case, guanine. Okay? That's a nucleotide. As a result, we get this long, thin molecule that resembles a ladder with alternating phosphates and sugar on the backbones and paired nitrogenous bases as the rungs. All right? I'm going to show you a slide. This is the way it describes it pretty good. And you've seen this diagram already, except you saw it in color. Now it's in black and white. All right? Again, you have sugar, phosphate. You have a nitrogenous base, a T with an A, a G with a C. All right? Now, you're used to seeing it twisted. And what you see over here, that's a great representation of DNA. If you took this right here and just twisted it, all right, you would end up with what you see on the right-hand side over here. Unfortunately, that is not a real good representation of DNA. It's because it doesn't go across the middle. This diagram actually is a little bit better. And no, I don't expect you to know this. I just want you to understand, when you see that twisted ladder that I just showed you, when you see this over here, that's DNA. Go with it. But it's not really accurate. This is more accurate. And you can see where here's the phosphates, here's the sugar right there, and then you have the nitrogenous bases that go at an angle. So you have, again, the phosphates here, the sugar here, and you have the nitrogenous bases that go at an angle. But that opens up a large area over here, and a small area over there, and a large area over there, and a small area over there, which is why we have this different shaped grooves. Why, if it's drawn really correctly, you're going to see something that looks... That was terrible, Williamson. Let's try that again. Much better. You're going to see something that's offset. Do I expect you to be able to draw a DNA molecule? No. I just want you to recognize that there is a difference between this, the twisted one where you just make a spiral, and this offset one, because this is more accurate. And there's a need for that, because we need to be able to get like enzymes and things into here to do some of the things that we're going to talk about with DNA, is that large groove. Okay? So how does the structure work? Well, to work, I've got to be able to explain to you how it stores information, how it's able to be copied, how it's able to make mistakes, and how we can read it. But it all comes from that spiraled staircase, that series of nucleotides of sugar, a phosphate on the outermost rungs, sugar, phosphate, sugar, phosphate, sugar, phosphate, antiparallel and complementary. And the corresponding nucleic acids, the T's with A's, the G's and C's, adenine, thymine, guanine, cytosine. And you need to know the structure. You're going to need to know what has one ring, what has two rings, which has two hydrogen bonds, which have three hydrogen bonds. But I've got a way to help you. You've got to help me out, though. You got to make sure that you understand A's with T's, G's with C's. If you can do that, I can get you, without you even thinking you know it, to remember which has two rings, which has one ring, which has two bonds, which has three bonds. So real quick, let's take a quick review. Watson and Crick, like in the first video, they knew that it was large, long, and thin. They knew that the size of the things, they knew it was a sugar, a phosphate, and a nitrogenous base. They knew it had to be able to store information, be able to be read, be able to copy itself, be able to make mistakes. They knew the sizes of purines and pyrimidines, and they knew that 1-to-1 ratio of adenines with thymines, guanines with cytosines. And what they came up with was that spiral, that double helix. That's what a helix is. Hela means spiral. That's where we get the word helicopter. It's a spiral. Okay? They came up with a double helix. What I'm going to do over the next few videos is explain to you how it does all those things and how we get protein from DNA. We don't get really protein from DNA. We get the information how to make protein from DNA, protein synthesis. If you have any questions, make sure you ask in Edpuzzle or ask me in class. Go back, look at your notes, see if there's any spots in it that you need some to fill in. Go back and review the video if you need to. Ask me questions in class. I hope you took good notes. Good luck.